Everyone for joining us today. The lecture we have today in this series is Pediatric Brain Cancers as Epigenetic Diseases. It's being presented by Stephen Mack. Thank you very much, Stephen, for presenting here today. He's an assistant professor at Texas Children's Hospital and Baylor College of Medicine. And his team studies a variety of high-risk brain tumors to understand the role of deregulated epigenomes as drivers of cancer. Stephen is also a recipient of one of Alex's Lemonade's most prestigious grants, and it's an ALSF A award, which he received in 2018. Dr. Mack's lecture will discuss developments in our understanding of chromatin regulation as drivers of pediatric brain cancers and therapeutic vulnerabilities. This is the fourth lecture in this virtual series. We have many more lectures scheduled in the coming weeks and months. If you'd like to be notified or see the topics of upcoming lectures, we'll put a link into the chat where you can see those, or you can sign up to be notified when new lectures are announced. We're also going to put a survey link in the chat so you can recommend topics and speakers for future lectures in this series. Finally, if you have questions for Stephen, put them into the chat and we will have them answered at the end. Thank you very much, and we appreciate you coming here today, Stephen, and everyone for joining us. And with that, I'll turn it over to you.
Thanks for the opportunity, and thank you everyone for joining me for this lecture at lunchtime. Let me go to my screen. Can you see my PowerPoint?
Yeah, we can.
Great. Thanks again to Jay, Alex Lemonade Stand, for supporting our research throughout the years and this opportunity to present today. I'll be talking about pediatric brain tumors, as I said, mentioned in my title, epigenetic diseases. More specifically, how they are fundamentally distinct from how we think about adult cancers. And in this slide showing, this is one of the first concepts that I learned when I was, took one of my first classes in genetics, the genetic basis of cancer, showing how in the case of colorectal cancer, you can have a normal epithelium in a colon crypt here, and showing how through accumulation of numerous mutations and increase in genomic instability, you can transform a cell shown in purple to this teal one shown in representing cancer here. So the thought is that if you could identify all the mutations within a specific tumor or cancer type, you'd be able to learn more about the mechanisms driving that specific tumor in terms of initiation, and also what are the specific driver events that are maintaining the tumors that could be potentially druggable, and what are the different alterations that are associated with therapeutic resistance. And this led to large-scale efforts by numerous consortia to catalog the different mutations, DNA alterations in cancer. And this was largely to work towards an effort to develop personalized cancer therapy. And what I mean by this is that, say you have a patient A with a specific tumor mutation, you could potentially match this gene mutation with a drug targeting the specific protein. And this is also true for patient B with a different alteration that you may be able to pair with a specific drug B. And in order to develop more safer, more effective therapies versus conventional drugs that are used potentially in children, which have a lot of cytotoxic effects. And there's been tremendous success in this area, largely looking at proteins at the cell surface, many of them representing kinase proteins. Number one, because these were largely accessible at the cell surface, and many of these were enzymes. So we had experience in designing small molecules and fitting molecules within these specific proteins for inhibition, such as inhibition of EGFR signaling pathways, HER2 in breast cancer, TRK receptor signaling in a variety of cancers. And I'll just highlight one of these success stories, or early success stories, in the case of personalized targeted therapy, in the case of tumors with recurrent fusions of the TRK receptor here. Just one second. The TRK receptor. And what this pathway is, it's an oncogenic pathway driving proliferation and cell survival, but small molecules have been developed against these specific fusions. And what I wanted to highlight in this right panel here is number one, the response, or the decrease in tumor size in response to this therapy, but also shown in the different colors are the different entities or tumor entities in which we're seeing response because they have the same specific driver. And then this is really exciting, and there's been additional examples, particularly in low-grade gliomas, using MAP kinase inhibitors. But what was one of the challenges that emerged when analyzing a lot of these large-scale sequencing cohorts by many groups is that compared to adult cancers shown on the right, pediatric tumors had very low, relatively low mutation rates, and these are number of mutations per megabase here as shown in the left. So this posed a challenge in terms of the number of targets that we could, effective targets that we could drug in terms of specific therapy. And one of the other challenges that we had was many of these proteins that were recurrently mutated weren't our typical receptor tyrosine kinases that were on the surface. They weren't proteins that we already had drugs for. These were, many of these targets that were recurrently mutated were an emerging class of epigenetic regulators. And what do I mean by epigenetics? So if you think of genetics as the DNA code, epigenetics for the purposes of this talk, although I'm quite simplifying this, is that these are the proteins that are regulating the transcription or the activation of these genes. And this occurs through multiple layers of modifications, proteins that bind this DNA, proteins that wrap this DNA, and generally how it's folded within the nucleus. Now, turning to normal development, the way I think about this is that every single cell in your body has the same DNA genome or genetic code, but you have hundreds of thousands of different cell types. And this is all governed by different transcriptional programs and regulated by different epigenetic mechanistic pathways. So for example, this is what distinguishes a kidney cell from a pancreatic cell, from a colorectal cell, from a brain cell. And there are numerous levels of this epigenetic regulation. So if you imagine here, within every cell, you have a 2-meter piece of DNA that needs to be compressed within a small nucleus that's less than 0.006 millimeters wide. And this has to be tightly regulated. And the DNA itself can be modified with the addition of, let's say, methyl groups shown in black here. This DNA is further associated with proteins known as histone proteins, and it's wrapped here in these nucleosomes. And these actual histone proteins can be modified themselves. So the way I like to think about this is these had flags that indicate whether a gene could be expressed or repressed. And for the purposes of this talk, when I mentioned we're going to focus on a lysine 27, histone 3 lysine 27 residue here, that when it's acetylated, we'll be referring to it as activated, and when it's methylated, it's repressed. But several of these regions can be further organized into neighborhoods. So the blue neighborhoods here containing these genes don't physically interact with these red neighborhoods shown here. And these are further organized within the cell, within the nucleus. So this is a highly intricate process of gene regulation known as epigenetics. Now, developmental biologists have been interested in this and studying it for some time. And so as shown here, let's say, for example, a gene that it's expressed and enhancer regions that are located further away from this gene. And remember that DNA is not existing as a single line, but it's actually physically folded. So these regions are potentially interacting with this gene here. And what they're doing is recruiting proteins known as transcription factors that can govern gene expression, govern when a gene is expressed, where is it expressed, and these processes are critical for organismal development. Now, in pediatric cancers, as I mentioned before, these are what was found from many of these large-scale sequencing studies is that there's an enrichment in mutations of epigenetic regulators. And I know this is a busy slide, but how I like to think about it is in terms of open chromatin, so actively transcribed regions of the genome that are driving oncogene expression. These are genes important for driving, let's say, cell proliferation and tumor growth. These are regulated by proteins, as I mentioned, transcription factors, let's say, that are activating these specific regions. And these are associated with active chromatin marks, such as lysine 27 acetylation. Conversely, there's regions of closed chromatin, which are silencing tumor suppressor genes, so genes involved in inhibition of cell growth and tumor formation. And what I wanted to highlight from this is many of these epigenetic proteins that regulate these processes in terms of activating gene expression or repressing genes, many of these epigenetic modifiers are mutated in pediatric brain tumors. MLL2 and 3 in medulloblastoma, SETD2 mutations in pediatric glioma. The histone mutations themselves, and I'll get into this further in the talk, they're mutated at that lysine 27 residue. And these govern or are thought to contribute to aberrant gene expression programs within that are involved in brain tumorigenesis, pediatric brain tumorigenesis. So as I mentioned before, I like to think of these modifications as on or off switch, and many people like to refer them as a dimmer to turn a gene expression on or off, but I like to think of it as one of these smart lights in terms of these proteins regulating when a gene's expressed, how much is it expressed, and where is it expressed. So this is a highly coordinated process that gets aberrantly regulated or deregulated in the case of pediatric cancer. So one of the central focuses in my lab is on cancer epigenetics and the role of epigenetics and cell identity. And this, as I mentioned before, is contributed through genetics, but there's other factors that contribute to epigenetic programs, such as metabolic factors, what are the nutrients available to cells, microenvironmental factors, what are the cell-cell interactions between tumor cells and other cells in the microenvironment. And in normal development, these programs regulate, let's say, a neural stem cell and how it changes or differentiates to a neuron or a glial cell. But these programs are also actively involved in transformation from a neural stem cell, a more differentiated cell, to a brain tumor cell. And there's also heterogeneity within these specific programs that distinguish between types of brain tumor cells and their tumorigenicity. So for the purposes of this talk, I like to highlight the role of epigenetics and their important contribution to pediatric brain tumors. And I'll do this through a couple examples. The first one highlighting the application and utility of learning about epigenetics and applying this to study and diagnose pediatric brain tumors, what mechanistic insights we can learn from studying the tumor epigenome in pediatric brain cancers, how these processes initiate tumorigenesis, and whether we can derive any therapeutic vulnerabilities from learning about the tumor epigenome, with a focus on pediatric brain cancer. So the first topic I'd like to highlight is DNA methylation, a modification that I mentioned earlier to DNA as an effective molecular classification tool. Now, my lab studies a variety of pediatric high-risk brain tumors. One of these is ependymoma, and I'll use this as I'm working through some of the examples in this lecture. But initially, ependymoma, it's the third most common pediatric brain tumor. And it arises commonly in infants, but it also can occur in adults. It arises across the entire neuroaxis, and the key thing for ependymoma is that, particularly in children, these are highly aggressive tumors. They're treated by surgery and radiation, which has significant long-term neurologic side effects. And there's a desperate need to learn more about this particular brain tumor to devise targeted, more effective therapies. Now, when I first started learning about ependymoma in Michael Taylor's lab in Toronto, this is what we knew about it. It was divided into various histological grades, grade 1 versus grade 2 and grade 3. Grade 1 tumors, as you can see here, have very distinct morphological features. But what was the challenge was distinguishing between what was the role of histopathological grading in terms of assessing which were the more aggressive tumors for the ones that were less aggressive. And this was a challenge that was particularly problematic for the field in that we couldn't agree on any consistent criteria to grade these tumors. So what was performed, or what we turned to in the field, and this is performed by numerous groups now, is that despite histological similarity, ependymomas, when we profiled them using DNA methylation profiling here, what this indicated to us is that ependymoma, although histologically similar, these tumors are broken down into at least 9 different molecular subtypes. as shown with these different colors here. And I'll get into more detail about this after. But these histologically similar tumors that look very similar in the microscope have very different molecular biology. Now, this work was greatly expanded by Stefan Pfister's group, in which they've created a DNA methylation atlas of all brain tumors. And you can see here ependymomas, which are forming each of these different islands here representing a different tumor subtype. So you could see Raleigh-driven ependymomas here are different from PFA ependymomas and PFB. But what I wanted to highlight from this study is that this is a simplified map, but there are potentially hundreds of different rare pediatric brain tumor types. And the additional thing is that these molecular profiles could be used to predict an unknown sample. So if you had a brain tumor specimen that you're interested in what subtype this could be, you could use this data to objectively predict with confidence or a confidence score of what that particular brain tumor type or classification is. And the next topic I'd like to highlight is epigenetic programs and cellular identity as therapeutic vulnerabilities. And as I mentioned before, one of the challenges that when we are sequencing a lot of these tumors is that we didn't identify clear drivers that we could pair with a specific drug. So what do you do in these cases? And what can we learn about what makes an ependymoma cell an ependymoma cell? And can this be leveraged for a particular therapy? And what we turned to as part of our ALSF project was looking at specific genes that were highly expressed within ependymoma and that were governed by super enhancers. And these super enhancers are specific regions of the genome that are governed by an intricate interplay between specific transcription factors. And these regions are heavily acetylated, so K27 acetylated. And this is an active mark associated with increase in transcription of these oncogenes, or in many cases, these super enhancers are known to identify genes important in maintaining or defining cell state. So shown in these two different cohorts, we identified these super enhancers that were present in ependymoma. But what we learned from these was the intricate transcription factors that were regulating these specific regions, that were binding to these regions. And many of these transcription factors that were highly active, as shown in red, are involved in normal development in gliogenesis. In an RNAi or loss of function screen, what we found was that many of these transcription factors that are potentially governing ependymoma cell identity are highly essential for cell growth, as shown here and within this specific screen. So what this indicated to us is that these particular proteins that are important or critical for maintaining ependymoma cell identity are required for proliferation or growth of these specific tumor types. Next, I'd like to highlight mutation of an epigenetic regulator as the first event in pediatric brain cancer. Now, one example of this is seen in pediatric high-grade glioma, in which there's a mutation of the actual histone proteins themselves. So as I mentioned, that these histones can be modified, and at this lysine 27 position, you have mutation of this histone to a methionine. And what this does is it blocks the PRC2 complex, so effectively blocking the repressive mark or the off switch. So the next question, the next thought is, what are the active genes, or what are the genes that are being increased in terms of transcription as a result of the loss of this repressive signaling? And what's also important to mention is that these specific histone mutations as the first event are subsequently accompanied by other mutations such as TP53 mutations, HRX mutations, PDGF receptor alpha amplifications. Now, what we had shown with Nanda-Shibata was that these mutations are associated with widespread accompanying epigenetic alterations. And just to take a step back, what we learned was that in normal cell development, your cells have 2/3 of your genome is comprised of endogenous retroviruses or repeat elements, and these are normally silenced through evolution. But what occurs in the context of this mutation is that you have aberrant activation of many of these repeat elements. So if you expression of many of these endogenous retroviral elements that are normally silenced within each of your cell types. Now, your cells have an inherent mechanism for responding to infection, so RNA-encoded viruses, And in this case, this represented a potential therapeutic vulnerability in that you have cells with these specific mutations could potentially leverage this by increasing levels of these retroviral elements so that cells undergo what's known as a process called viral mimicry. So they think they're virally infected and respond by upregulating an interferon pathway to respond against these sequences. And we had shown with NADA that this represents a potential therapeutic epigenetic vulnerability in that tumor cells with this specific mutations can be further treated with these epigenetic inhibitors to further increase these levels of endogenous retroviruses as compared to a knockout control, demonstrating that this is a potential sensitivity to these specific inhibitors and is associated with a specific mutation in this histone protein. What about mutation of an epigenetic regulator as the only event in cancer? And there's a good example of this, not only in pediatric ependymoma, but many other solid tumors in pediatrics, such as EWS-FLI1-driven Ewing sarcoma, PAX3-FOXO1 fusions, and rhabdomyosarcoma. But we study a variant of ependymoma known as C11orf supratentorial ependymoma, that is ependymoma of the cortex. And these, over 72% of these tumors are driven by the C11orf95 REL-A fusion. And this is identified by Richard Gilbertson's group and David Ellison. So this is a fusion between an uncharacterized protein called C11orf95, chromosome 11, open reading frame 95, with the well-characterized REL-A pathway that's been shown to be involved in inflammation and cancer. What this fusion does is it traps or drives this specific protein into the nucleus. And what's important to, what I'd like to convey here is that this fusion alone is sufficient to driving tumorigenesis. So I say, to compare this against many of the adult tumors that I alluded to in the beginning of the talk, this is a fusion, one event that's sufficient when driven in a specific neural stem cell or neural cell type that can cause cancer. And this is shown here in the mice that are developed ependymoma-like tumors that are driven by this specific alteration. And we had also shown this in using other approaches in collaboration with Ben Deneen, in which we overexpress this specific fusion in utero during development, brain development. We're able to generate these tumors. This fusion is able to localize to specific regions of the genome, as shown by CHIP, seq, or cut and run of this specific fusion. And it's associated with the acquisition of active chromatin. So these activate specific regions of the genome that are driving specific oncogenes such as FRNB2, cyclin D1, that are known drivers of ependymoma. And the last example I'd like to give is the case that epigenomic alterations may be occurring in pediatric brain cancers, may be drivers without accompanying DNA mutations. And we were studying this in the context of sequencing ependymoma by whole genome sequencing and whole exome sequencing. And what was immediately apparent about these tumors as compared to adult, let's say adult lung cancer genomes, is that there's many different alterations in lung cancer, a few recurrent rearrangements, copy number alterations, recurrent mutations. But this is what we saw for a large number of infant posterior fossa ependymomas, that is ependymomas that are arising in the cerebellum and brainstem, is that it's almost like we sequence the normal genome here, that there's a lack of recurrent mutations, there's a lack of recurrent rearrangements or copy number alterations. Ependymomas have a very low mutation rate. Now, what was shown by David Ellison's group was that these specific tumors have overexpression of a gene known as CXOR67, and in about 10% of cases it's mutated, but in 90% of cases it's overexpressed. And this protein acts as a mimic of the K27M mutation, as I alluded to in earlier slides. But overexpression of this particular protein is sufficient to turn off that repressive mark, drive down K27 trimethylation. And this specific epigenetic phenotype we see in pediatric glioma, it's a hallmark of a PFA ependymoma, including pediatric glioma, that are driven by the K27M mutation. But the difference between gliomas and ependymomas is that these are accompanied, as I mentioned before, with additional alterations, additional mutations. And many ependymomas that we see, PFA ependymomas, have overexpression of this CXORF67 protein or EZH2 interacting protein is sufficient to drive down K27 trimethylation levels or loss of the off switch to activate gene expression programs. And that's often the only thing we see in these specific tumors. So the last thing I like to leave with the thought is that epigenetic programs play a critical role in differentiating different cell types in the body, let's say neural stem cells from neurons, glial cells. But who's to say that specific epigenetic programs could play a primary role in driving tumorigenesis in the case of pediatric brain tumors? I think there's a lot more work to be done, and perhaps we haven't identified cryptic genetic alterations yet in these PFA-driven tumors. But what I'd like to highlight in this final slide is that shown in the inner circle, the different discoveries we've made using DNA-based approaches, discovering drivers of many different pediatric cancers, and how these are making their way in the clinic as shown in the outer circle. I think there's a lot more discovery to be done in terms of learning about the role of epigenetic programs in a variety of pediatric cancers, not only brain tumors. And these have important ramifications in terms of new technologies that we can bring to the clinic, such as DNA methylation in the case of pediatric brain tumors. With that, I'd like to thank my team and all my collaborators and our funding agencies, Alex Lemonade Stand, for giving me this great opportunity to present today. And thank you for your attention.
Thank you, Stephen. That was a great lecture. At this time, we're willing to take some questions. You can just type them into the chat if people have questions. We'll give it just a minute to see if we get some questions in, Stephen. Looks like somebody has their hand raised. When I tried to do it last time, they had themselves muted, so it didn't come out. But I can't figure out how to click on it today. Jen, are you able to? Okay, here we go. I'm going to start reading these. From Hassan Uladag, how likely is it to develop a drug for rare brain tumors?
That's a great question. I think that as we study these brain tumors more and divide them into different subtypes, we see these specific alterations, let's say the C11orf95 REL-A fusion that's seen specifically in a subtype of ependyma. I think that in terms of partnership with industry, I think that there's potential there to develop specific molecules for this specific protein. I think that what I think that I like to focus on is that we have a target, we have a specific driver, so we can isolate the patients if we had a specific drug that we could leverage for that particular tumor type. But I think that it requires partnership not only from academia in terms of the initial development of this, but partnership with other groups as well, including industry, to take this to an actual development of a specific drug.
Thank you. Next question is from Jerome Fortin. His question is related to DNA methylation. Do you think it's useful only as a classifier of tumors, or does it drive certain aspects of tumorigenesis?
That's a great question. I mean, initially when we were profiling DNA methylation, we had our thought at that time was that, or at least the field is that when you have hypermethylation of genes, it's associated with gene silencing. And I think that's true for some cases where you have repression of tumor suppressor genes. But I think that in many cases, the utility of it is to use it as a marker. I think that in the case of pediatric brain tumors or even sarcomas, it's a reflection of the patterns that are present in the cells that give rise to these tumors. And it has power in terms of use as molecular diagnostics. I think that there's also potential for utilizing specific inhibitors of these DNA methyltransferases in cases as a specific therapy and for inhibiting these specific programs, such as these endogenous retroviral element upregulation that we see in subsets of these high-grade gliomas and ependymomas, for example.
Thank you, Steve. The next question comes from Genevieve Kendall, and Genevieve is giving our next talk actually on Friday about using zebrafish models in pediatric sarcomas. So don't forget to sign up for that one. But Genevieve says it was a great talk. In your data, CXORF67 is also overexpressed in germ cell tumors. Do you see a similar mechanism of action at play in that context?
We haven't looked at that specifically. That's a great question. We have a few lines that I think we were thinking about looking at. There's a recent paper showing that CXORF67 is overexpressed in germ cells. So I think that it's very likely that we'd see similar mechanisms there.
Okay, sorry, I was muted. The next question is from UCLA, and it says, is there genetically engineered mouse models for ependymomas that could be used for drug screening?
There's a variety of genetically engineered. There's number one patient-derived xenograft models that are available, but there's increasingly more and more genetically engineered mouse models of supratentorial ependymomas, particularly driven by the REL-A or YAP1 fusion. That could be used for drug screening in the context of, so number one, you'd be able to grow those cells to screen them in vitro, and you'd have an immune competent model that you could test these specific therapies in vivo. PFA ependymoma models, I think we're limited to PDX models currently, as there hasn't been a genetically engineered mouse model for PFA ependymoma as of yet.
The next question is from Gustavo, and his question is for EP1NS RELA fusion line culture. It was performed under NS conditions or attached conditions. And follow-up, does it alter the epigenetic K27 AC signature?
So that's a great question. So one of the challenges when we are growing some of these lines in culture is that we're growing them in addition, how representative is that of the actual in vivo environment, and how reflective is that of the primary patient tumor? In the case of EP1S, when we've profiled it by DNA methylation profiling, K27 acetylation profiling, and we've compared it against the primary tumors, it retains that epigenetic signature surprisingly quite well. I'm not sure if there's other aspects that make it different, but at least looking at those two signatures would indicate to us that it reflects at least some of the biology of the actual primary patient specimen.
Okay, and the next question is from Andrea, I'm not sure, Punti. Says, fantastic talk, and her question is about CXORF67 and H3K27M. And the question is, do you believe that loss of H3K27 ME3 is the main route through which these proteins drive tumorigenesis?
I think that's a great question. I think that in the case of PFA ependymoma, these tumors typically have very silent genomes. They're very frequently devoid of copy number alterations. CXORF67 and their mutational overexpression is one of the only things that at least we've detected so far in these specific tumors. Do I think it's a major driver in the case of PFA ependymoma? I think quite possibly. I think that we could probably test that in some of our models as well. In the case of pediatric high-grade glioma, I think that given that there's accompanying mutations, that would suggest that there are additional hits. But the problem with, I think, PFA ependymoma is that I don't know what those additional hits would be.
I don't know what all these letters and numbers mean. I'm glad you do.
At least I think I do.
Okay. So the next question comes from Bernard Englinger, and he's also said it was a great talk. And he said, what is your thought that one expression of non-canonical NF-KB target genes in RELA EPN?
So I think that's a great question. I think that From our data, what we found is that there are, and this is in line with Richard Gilbertson's work, in that there is a shared program of canonical REL-A target genes that are overexpressed. But there is a distinct signature, gene expression signature, and these are bound by the actual fusion itself. So I think that connecting C11orf95 with REL-A confers a novel gene expression program that's different from hyperactive REL-A signaling. And also add to that is that Eric Coughlin has shown that when you drive expression of hyperactive REL-A, that's not sufficient to drive tumorigenesis in mice. So it suggests that there's something additional, something added that C11orf95 is providing.
Okay. Next question is, how does your group plan to handle issues with testing ICI strategies in combination with HDAC inhibitors in the absence of a genetic mouse model with an intact immune system?
That's a great question. I think that we're transitioning now from a lot of our PDX models, given that we are exploring the role of immune cell recruitment into immune competent genetic models. And I think this is where a lot of our efforts in terms of in utero electroporation approaches are at play here, or an application that we can use that specific mouse model. Probably can't do it for PFA and pentamoma, but I think that we could test that potential strategy in the case of pediatric high-grade glioma with that specific histone mutation.
So the next question comes from Brian Na, and it's a multi-part question. It's pretty long. So I'm going to give it a chance to read it to you, Stephen. And he says, what do you think about the role of the epigenetic modifying agents, such as EZH2 inhibitor? For example, tazemetostat is now in phase 2 studies within the COG match, but a recent paper on ATRT out of Heidelberg shows that EZH2 and H3K27 ME3 and EZH2, I guess, positive and negative, which suggests that an EZH2 inhibitor could, in fact, may have a paradoxical effect, although the phase 1 trial was promising. So what do you think the next steps would be for that?
I think that's a great question. If I'm understanding correctly, I think that given the evidence out there that it's K27 trimethylation, there's different ways of tackling it. You can restore K27 trimethylation levels, but one approach is that even though there's a global loss, we see a global loss of K27 trimethylation, it's retained at specific sites, and it's thought that these sites are to be important for repression of tumor suppressor genes. So there's a thought that that retention of that small amount of K27 trimethylation could be driven down further as a therapeutic vulnerability with EZH2 inhibitors. So I think that there's potential there, and I think that there's rationale for at least testing this in preclinical models that have this epigenetic phenotype of low K27 trimethylation.
All right, thanks, Steve. Next question also says, great talk, Dr. Mack. Have you looked at the epigenetic profile of non-RELA fusion ependymomas, for example, the YAP1 fusion tumors?
We haven't looked at them directly, but I think that, that's a great question as to how we compare the fusion-driven transcriptional programs with REL-A to YAP1. For those of you in the audience, YAP1, which I didn't discuss in this particular talk, it drives a particular subtype of ependymoma that's distinct from REL-A. But it'd be interesting to see what are the commonalities between C11orf95 REL-A-driven tumors and YAP1 fusion-driven tumors, particularly C11orf95 YAP1-driven ependymomas.
Okay, the next one comes from David Chen, and he asks, is there a cell model of ependymoma available to lentiviral-based CRISPR genetic screen for looking for new therapeutic targets in this disease?
That's a great question, David. I think that there's numerous PDX models that are becoming available now, particularly for REL-A-driven ependymoma and also PFA ependymoma. I think that we have a collection of models here that we're happy to share with anyone who's listening here, because I think that I think that it's a good idea for us to do these large genome-wide screens for looking at potential vulnerabilities, or even in a focused manner looking at epigenetic vulnerabilities in these specific tumor types.
Okay, the next question comes from Anna Green, our good friend. And she asks, why is ependymoma chemo-resistant, and how will you overcome that when looking for new therapies?
I think that's a great question. We don't fully understand why ependymomas are chemo-resistant, to my knowledge at the moment. I think that there's numerous avenues for why that might be the case. I think that exploring in terms of deep biology of what are the mechanisms driving that resistance, whether it be exporting drug pumps exporting from the cell or other mechanisms, I think that it's worth exploring those areas. I think that there's other approaches in terms of looking at more specific targeted therapies. I know that some of our colleagues here are looking at HER2 expression on ependymoma cells as a potential vulnerability, and targeting at ependymoma cells by virtue of high-level expressions of HER2. So I think that both exploring the mechanisms of contributing to chemo resistance, but also looking at more targeted, more specific approaches, I think would be valuable as well.
Well, we've reached the end of our questions, Stephen. So I want to thank you again for doing this talk today. I want to thank you for everything that you do to help kids with cancer. We appreciate it. And I would just thank everyone for coming to this lecture today. If you look at the chat, there's a survey there where you can give us feedback about how we're doing with the lecture series, and also recommend future speakers and future topics. So with that, I'm going to call this lecture a success. And thank you, Stephen, and thank you, everybody, for joining us.
Thanks, everyone.
Bye-bye.